Welcome back, Mindsetters. Remember that we are doing Life Sciences Grade 12 revision on both Paper 1 and Paper 2. I know that this is an awesome lesson because you guys are writing on this. And even if you have written, prelims are very close, and so are other tests that you may have. So I know that this is going to be awesome for that, and you're all going to ace it. I also want to remind you on the winter school that will be taking place from the 2nd of July to the 8th. And it is a boot camp. It is awesome. It is hectic. And you guys must be so ready for that. And remember that it's winter. So you must also keep warm and keep safe and stay indoors and study rather than going outside and getting yourself sick. And when you're indoors, make sure that you tune into channel 319 because it is an awesome, awesome winter show for us. Over to you, Aslam.
Thank you, Godlecho. Okay, guys, welcome back again. And we're going through with our genetics question. Just before that, we had a question earlier about viruses that was sent through to us, and the learner wanted to know, how do viruses live inside organisms when they do not have a nucleus? Very easy. Remember when you did viruses, by the way, the actual structure and functioning of viruses is from grade 11 and not for grade 12. But it's linked to DNA, so we will just answer the question very quickly for you. Remember that viruses do not have a nucleus, but they do have DNA or RNA, not both. And it is this DNA and RNA that enables them to reproduce more viruses when they get into your body or a body of any organism. The DNA or RNA is injected to a cell or cells inside the body, and the DNA and RNA replicate. They make more copies, and the cell which they infected then bursts, releasing more viruses there. They are considered to be non-living because they cannot respire, they cannot metabolize, they do not have mitochondria, etc. But once inside the living cell, they are able to reproduce, only using the DNA. I hope that answered that question. Let's move on. This question here is a useful investigative planning type of question which examiners have been seeming to favor in the last few exams. This particular one says a fingerprint is a useful way of identifying people and classifying them into groups. We know in South Africa, the minute you apply for an ID, as in any other country, your fingerprints are taken. So on the database, your fingerprint is already there. Your ID is registered to your fingerprint. So we can be classified according to that. Fingerprint is taken by rolling the right index finger on an ink pad and then onto a piece of paper. You probably, grade 12s, by now you should have your ID. Remember, you need your ID for the end of the year to identify yourself in an exam center. So you should have your ID. So you should have had this experience of taking your fingerprint or having your fingerprints taken. During a discussion of this topic, a group of learners asked the following question. Which one of the 5 main types of fingerprints is most common amongst the learners of this school? The 3 main groups of fingerprints, by the way, are either arced, whorled, and the last one I forget at this moment. I'll get back to you on that one. For some reason it's not coming to me, blank. I'll get back to you on that one just now. But these are the types of fingerprints that we have. And what they're saying is they want to know which one is most common. And they want to ask the first question, state any 4 steps in the planning process. State any 4 steps in the planning process. Now guys, planning is not restricted to science. It's not restricting to maths. It's not restricted to this particular experiment. Planning is planning. Wherever you do planning, whatever the function, whatever the job you have to do, the planning steps will be the same. If you are planning the matric farewell, that should be relevant to you right now, then you would firstly want to know how many learners you have in the matric farewell. You would also want to know where the venue you would choose. You would also want to know what is the menu, etc., etc. So these are planning steps. Now in this particular thing, we want to know what? We want to know what is the most common type of fingerprint. Oh, by the way, that thing came back to me. Looped. There we go. Arc, world, or loop. Those are the three main categories. And within these categories, there are other categories. By the way, mindsetters, don't get worried, because you do not have to know the fingerprints themselves. It is not part of your curriculum. If a question like this is given, then obviously if they need to know the names would be in the passage. So don't fret, don't drink too much of coffee, don't get an energy. Wow, I need some energy because I don't know these things here. No, it's not going to help you. It's not going to be a problem if you don't. These things are going to be given in the question. Okay, it happened to me on live show. I forgot, I got it back. And in the paper, same thing, guys. If you forget something while you are answering question 1.3.2, don't sit and say 132, 132. I can't get this in my head. I have to stay here. You're losing time in that time. Same in accounting. If you are balancing your books and your books don't balance, many learners waste minutes and minutes and minutes trying to balance the book. In the meanwhile, the balance itself is only 3 to 4 marks, and they leave the rest of the question blank. If you have this blank moment, move on with your paper, and at the end, you come back to this question, making sure that you left a space on your answer book for this particular question. So you know, as a reminder, that I haven't answered this question. Also, so that you keep parts of a question together. Because when markers mark, generally, the way marking is done is markers mark a particular question. So there's some team marking question 2, 3, 4, 5, etc. and they will mark that one. If you're going to put your answers all over the show, the guy marking question 2 may miss something. And then it will come back later from someone else and so on. So rather keep parts of a question together. So in this one here, obviously we're dealing with learners at a school. So first of all, we must determine the sample size. Putting it in scientific terms now. In other words, what we're saying here, guys, is how many people are we going to investigate? Are we going to investigate the whole school? Are we going to investigate only the grade 12 learners? Etc., etc., etc. So we must determine the sample size. Obviously, because of the nature of this particular exercise, and what you have to do, you have to use a rubber pad, an ink pad, and you're going to get that. So make sure you acquire the equipment. I'm writing in general terms, people, because in an exam you will not get exactly this question here maybe. It's some other planning. So any planning will require equipment. And in this case, you will specify. You will get paper and the ink pad, etc. Now, when you collect the results of any investigation, how are you going to present it? So then you're going to have to decide on how the data will be presented or collected and presented, etc. And obviously, just like the matric farewell, you'd obviously need to have the venue. Where are you going to do this? You can also add to this. There's 1,000 other things you can add. You can add, for example, determine who is going to be the person that's going to take the fingerprints. Who is going to record it, etc., etc. Logistical issues. So in other words, any planning, people, whether it's here or anywhere else, it's going to be the same. Okay? Provide a hypothesis. Now look at this question carefully. Provide a hypothesis for the question the learners asked. They don't want just any hypothesis. They want a hypothesis based on the question the learners have asked. Now, just before we go to the question that the learners asked, remember a hypothesis is a predictive or predicted statement. It is not a question. It is not a conclusion. It is not an aim. For example, aim, we want to know, to determine which particular fingerprint type is the most prevalent in this class or in the school. That's an aim. That's why we're doing this experiment. So it's not a hypothesis. Okay? And it's not a question. So the question we go back to, let's go to the question and show you what is a question again. Which one of the 5 main types of fingerprints is most common among the learners at this school? That's a question. So you can't repeat that question here because this is the question that these learners wanted to answer. Now, a hypothesis is usually done before the experiment is done. In other words, somebody is saying, you know what, I think X, Y, and Z. Not just thinking wildly, but based on what they think, what they know, their knowledge, etc. They're making a hypothesis. Okay? And if we go to that again, to this question, let's go to the question. So they want to know which type of the main types of fingerprints is common. In a hypothesis, we must look at the independent variable and the dependent variable and the relationship. That's why you get 3 marks for this. Because you must show the independent variable, you must show the dependent variable, and the relationship for it. That's why it has to be put in the form of a statement. You must take a stand. You cannot say which one is going to be more. No, you've got to say one of them. For example, we said earlier, we're looking at arced, world, looped. So any one of those words you could have used there, that is your independent variable. The arc, the world, the loop type, those are the different types. Is more prevalent among the learners, is most common among the learners of the school. So we're already saying is most common. Most denotes a comparison. You are taking a stand that this particular one, or this one, or this one, or whatever. You can say X, Y, Z, whatever in this particular answer is the most prevalent in the school. And remember, hypotheses are causal statements. That means these are because of that as well sometimes. For example, I may say, if I give her a diamond ring, she will marry me. Wow. If I give her a diamond ring, she will marry me. Now look at that. It's what I am predicting. It's not the truth. How do I figure that out now? I've got to go with the diamond ring and present it to her. She may take the diamond ring, turn around and run out of the restaurant and leave me high and dry. Or she may someone clap me right there in the restaurant in front of everybody. That wouldn't be a good thing.
Yeah, that would be a horrible thing.
Terrible, terrible, terrible. Or she might just say, yes, darling, I take you to be your lawful wedded wife forever and ever. That would be good, no?
Yeah, that would be
After
the
my
ideal.
investment of the diamond ring. So can you see, in a hypothesis, there are many ways in which after that something can happen. So the experiment is my actually taking the diamond ring and giving it to her. And after that, I must decide whether my hypothesis was correct or not. So remember, hypothesis, guys, is not something that is only restricted to science. Every single day, you and I, every single one of us is making a hypothesis of sort. If I take this road, I'll get there quickly. If I brush my teeth, I will not get tooth decay, etc., etc., etc. If I study hard, I will get a good result. These are all hypotheses that we are doing every day. So get in your mind one such hypothesis, and whenever you get a hypothesis question, try and fit that hypothesis question into your particular question, or hypothesis, sorry. This one here shows you a pedigree diagram of inheritance of flower color in a certain plant over 3 generations. Use the symbols for contrasting alleles. W, capital W, notice another clue here, capital W is given for white color and R for red color. Now, when I put this question up, something moved in the diagram, so I'm going to correct that first. This should be a red flower, and that should be a mixed red white flower. In other words, they're saying that both red and white show in this particular pedigree. Now guys, in a pedigree it's very easy. All you need to know is your work in terms of genotypes, etc., and your phenotypes. And in this case, you need to know the difference between complete dominance, incomplete dominance, and codominance. Complete dominance is when one particular characteristic covers or masks the other one. And we looked at an example just now with the gray coat and the white coat of those mice. The gray coat was the dominant feature, and therefore it masks out the white one. The white one could only show itself if the gray one was absent. I like to use the word, when the cats are away, the mice will play. So in other words, the dominant one is the cat, the recessive one is the mice. When the dominant one is there, you cannot see the mice, you cannot see the recessive one. When the cat is not there, the dominant one, then the mice will appear. Therefore, you'll see when you get a recessive characteristic like the other one we looked at, GG, only when that showed up we got the white. Before that, we did not get the white. Now let's look at this diagram. First of all, they're telling you that we're making red and white, and you notice you're getting something in between there. You're not getting red, you're not getting white, but you're getting something new, a red and white flower. So that tells you that these two characteristics, red and white, are equally dominant. So this is an example of codominance. Please understand that. In a case of incomplete dominance, you won't get a mixture of red and white. You will get a new color. For example, if we had red and white here and we get pink, that means that these two colors are neither of them are dominant over the other, and they have now formed a new blend. So please watch that when you are doing that. Type of inheritance, codominance. Explain your answer. Because both characteristics showed themselves equally when this was crossed. Last one there, use the symbol RR, R and W to write down the genotypes of each. In the case of A, let's look at A quickly. A is red, B is the mixed color, and C is the white. Okay, let's go back. Red will be R, B was the mixed one, so it will be capital R, capital W, and C would be capital W, capital W. Remember, in this case, both characteristics are used in capital because both are dominant. Over to you, Katlego.
Thank you. Guys, we'll be shooting over to a very quick break. This is for you to get all your questions that you may have, or maybe queries, or maybe you just didn't understand a little part that happened. Let us know during this break so that after the break, we can cover anything that you may need. See you after the break.